Hi everyone. So this is the third of my screencast videos on photosynthesis, and in this one we're going to focus on the second big part of photosynthesis called the light-independent reactions, also called the Calvin cycle. So just a quick review from my last video on the light-dependent reactions. We can think of these as the photo part of photosynthesis, the part that requires light, that occurs in the thylakoid membrane requiring sunlight. And this is the process that's going to generate this useful energy molecule, ATP, that's providing the energy for the Calvin cycle. And it's also generating NADPH, which is this activated electron carrier that's going to carry the electrons to reduce the carbons in CO2 and make them into useful sugars. So the second phase we're focusing on in this video, the light-independent reactions, we can think of as the synthesis part of photosynthesis, because this is the part that's actually making the sugar, synthesize, make. So here we are in this empty space in the chloroplast called the stroma. This process does not directly require sunlight, but it does require carbon dioxide from the air, and that carbon will be used to make sugars, carbohydrates. So here we go. What I'd like to do first is show you guys a TED Ed video that I think does a really good job of kind of animating, creating some analogies to help you understand this cycle. So let's start there.
You're facing a giant bowl of energy-packed carbon crunchies. 1 spoonful, 2, 3. Soon, you're powered up by the energy surge that comes from your meal. But how did that energy get into your bowl? Energy exists in the form of sugars made by the plant your cereal came from, like wheat or corn. As you can see, carbon is the chemical backbone, and plants get their fix of it in the form of carbon dioxide, CO2, from the air that we all breathe. But how does a plant's energy factory, housed in the stroma of the chloroplast, turn a 1-carbon gas like CO2 into a 6-carbon solid like glucose? If you're thinking photosynthesis, you're right. But photosynthesis is divided into 2 steps. The first, which stores energy from the sun in the form of adenosine triphosphate, or ATP. And the second, the Calvin cycle, that captures carbon and turns it into sugar. This second phase represents one of nature's most sustainable production lines. And so with that, welcome to the world's most minuscule factory. The starting materials? A mix of CO2 molecules from the air and pre-assembled molecules called ribulose bisphosphate, or RUBP, each containing 5 carbons. The initiator? An industrious enzyme named Rubisco that welds 1 carbon atom from a CO2 molecule with the RUBP chain to build an initial 6-carbon sequence. That rapidly splits into 2 shorter chains containing 3 carbons each and called phosphoglycerates, or PGAs for short. Enter ATP and another chemical called nicotinamide adenine dinucleotide phosphate, or just NADPH. ATP, working like a lubricant, delivers energy, while NADPH affixes 1 hydrogen to each of the PGA chains, changing them into molecules called glyceraldehyde 3-phosphates, or G3Ps. Glucose needs 6 carbons to form, made from 2 molecules of G3P, which incidentally have 6 carbons between them. So sugar has just been manufactured, right? Not quite. The Calvin cycle works like a sustainable production line, meaning that those original RUBPs that kicked things off at the start need to be recreated by reusing materials within the cycle now. But each RUBP needs 5 carbons, and manufacturing glucose takes a whole 6. Something doesn't add up. The answer lies in one phenomenal fact. While we've been focusing on this single production line, 5 others have been happening at the same time. With 6 conveyor belts moving in unison, there isn't just 1 carbon that gets soldered to 1 RUBP chain, but 6 carbons soldered to 6 RUBPs. That creates 12 G3P chains instead of just 2, meaning that altogether 36 carbons exist, the precise number needed to manufacture sugar and rebuild those RUBPs. Of the 12 G3Ps pooled together, 2 are siphoned off to form that energy-rich 6-carbon glucose chain, the one fueling you via your breakfast. Success! But back on the manufacturing line, the byproducts of this sugar production are swiftly assembled to recreate those 6 RUBPs. That requires 30 carbons, the exact number contained by the remaining 10 G3Ps. Now a molecular mix and match occurs. 2 of the G3Ps are welded together, forming a 6-carbon sequence. By adding a third G3P, a 9-carbon chain is built. The first RUBP, made up of 5 carbons, is cast from this, leaving 4 carbons behind. But there's no wastage here. Those are soldered to a 4th G3P molecule, making a 7-carbon chain. Added to a 5th G3P molecule, a 10-carbon chain is created. Enough now to craft 2 more RUBPs. With 3 full RUBPs recreated from 5 of the 10 G3Ps, simply duplicating this process will renew the 6 RUBP chains needed to restart the cycle again. So the Calvin cycle generates the precise number of elements and processes required to keep this biochemical production line turning endlessly. And it's just one of the hundreds of cycles present in nature. Why so many? Because if biological production processes were linear, they wouldn't be nearly as efficient or successful at using energy to manufacture the materials that nature relies upon, like sugar. Cycles create vital feedback loops that repeatedly reuse and rebuild ingredients, crafting as much as possible out of the planet's available resources. Such as that sugar, built using raw sunlight and carbon, converted in plant factories to become the energy that powers you and keeps the cycles revolving in your own life.
All right, so that goes into a little more detail than I need you to know about the Calvin cycle, but I think it helps to clarify the cyclical nature of this. So, key takeaways that I want you to feel comfortable with the light-independent reactions. These are also called the Calvin cycle. Right? It occurs in the stroma, and it can occur 24 hours a day. Often the highest rates of the light-independent reactions actually happen in the dark after a large amount of ATP and NADPH has built up inside the chloroplast. Carbon dioxide is going to enter the chloroplast, right? And it's going to combine with ATP and NADPH from the light reaction in order to form sugars. And at the end of this process, we basically can think about how we kind of have 2 empty batteries, right? We have an ATP that's been converted to its low energy form, ADP, and we have an NADPH that has dropped off its energized electrons and its hydrogen and turned into its sort of deactivated NADP plus form. And both of these empty batteries are going to be sent back to the light-dependent reaction to be re-energized, yet another cycle in this process. Okay, so we're going to divide this into 3 phases to help our understanding: carbon fixation, reduction, and regeneration of the RUBP. So let's look at each of those briefly. Okay, carbon fixation comes from this idea that we are going to be affixing carbon onto RUBP, right? Or as the video said, we're going to give RUBP a carbon fix, right? And so the incredible enzyme that does this is Rubisco. It is the most prevalent enzyme on Earth. And what Rubisco does, it has the amazing ability to take a carbon dioxide molecule and form a bond between the carbon and the carbon dioxide in this 5-carbon RUBP molecule, right? Forming, as a consequence, a 6-carbon molecule that you don't need to memorize the name of. Then, right, well, let me give you a moment. Go ahead and take a moment to pause the video and summarize that. Okay. Next is the reduction step, and this is reduction in the sense that we learned with redox. So literally, we are going to reduce the carbons in this 6-carbon molecule by adding electrons to them. And where do those electrons come from? NADPH. These are the electrons that originally came from splitting water way back at the start of that light-dependent reaction process. So our 6-carbon molecule is going to break apart into 2 3-carbon molecules. You don't need to memorize the names of these because this is actually a very unstable molecule. So it's going to just naturally break apart. And then what's going to happen is ATP is going to provide the energy for this conversion, and NADPH is going to provide the hydrogens and the electrons for this process. And through a series of reactions powered by these molecules, these three carbon molecules are going to be converted into something called G3P, which is essentially a sugar precursor. It's just half of a glucose. Remember, glucose has 6 carbons, so essentially 2 G3Ps are very quickly and easily converted into a glucose. Sometimes they'll also be used to build other essential molecules for the plant's tissues as well. The whole point, though, is in this form, carbon is useful. It is a useful energy source because it is reduced, right? So this is reduced carbons in the form of G3P. And then remember, let me give you a moment to process. So pause the video, give this a try. Okay. And then remember, in order for this to be a sustainable cycle, we have to have this third regeneration step. And in this regeneration step, if we think about this process happening in 6 different places simultaneously, because there are so many rubiscos floating around in the stroma of the chloroplast. So this is happening 6 times simultaneously, right? And it turns out that sort of 5 out of the 6 times, these G3Ps are actually just being used to regenerate RUBP. And 1 out of the 6 times, that G3P is going to go off and make glucose, right? So all I need you to know, I don't need you to track the numbers in super detailed ways, but I do want you to be aware that there's going to have to be cycling back. Most of this G3P is going to be used in order to regenerate this 5-carbon molecule that is essential for affixing CO2 too. And a smaller amount of the G3P is going to go off and be used to make glucose, okay? All right, pause the video, take a moment to process that. All right, so I want to take a moment before we wrap this up to track the atoms. Think about where the atoms started and where they ended. So let's think first about our carbon atoms. Remember that the carbon in CO2 has now been essentially affixed and reduced and become part of glucose, these C6, right? So these 6 carbons from 6 CO2s are now C6 in a glucose. The hydrogens, remember, have made a very long journey. Those hydrogens were originally part of water, which was split by photosystem 2. Those hydrogens passed through ATP synthase and were used to help make ATP. They were affixed to NADP+ to make NADPH, which eventually delivered them to the Calvin cycle, and they eventually got attached to these carbons as part of glucose. And then these oxygens, so interestingly, these oxygens are coming from a couple of different places. They're coming, so these particular oxygens are mostly coming from carbon dioxide, right? And then an interesting thing is some of these carbon dioxide oxygens are also actually getting released as water. Because remember back in that first video, in the net reaction for photosynthesis, we're actually making a little water as a waste product in the Calvin cycle. Right? So that's what happens to all these CO2 atoms, right? We've accounted for this this hydrogen in water. And then remember that the oxygens in water are all getting released as oxygen gas, which although it is a waste product for plants, is an essential component of our atmosphere and has made our Earth's atmosphere habitable for organisms like us in a way it was not before plants became common on Earth. Okay, I want you to take a moment on your notes now to review that and to try filling out this table on your notes. So pause the video and give this a try. Okay, and then I'm just going to put this up so you can check yourself. We'll talk about this more in class, but just use this as a reference as you're reviewing the key processes of photosynthesis.